today my lecture in soil microbiology is about the culturable and non-culturable prokaryotes. As you know, soil is a very complex medium for microbial growth. It contains a huge diversity of populations ranging from prokaryotes to eukaryotes, from bacteria to animals, including plants, fungi, algae, and many other things. When I say it's very complex, it means that the soil is highly variable. Its environment changes just because of its complex structure. So moisture is a very big factor also, because moisture also changes at very small distances. So it means that when we are going to study something in soil, we have to take care of all these things, that we have to cover up the complexity within that. So today's lecture is about prokaryotes, and prokaryotes are something, the simplest type of organisms on this in this. So prokaryotes are of two types, that is the bacteria and archaea. This is a tree you can see here. You can see three types of organisms: bacteria, archaea, and eukarya. In this tree, it's a very simple tree that is based on small subunit of ribosomal RNA gene. So in this, there are few groups of bacteria that are visible, and same for archaea and eukarya. So in this tree, these two organisms, bacteria and archaea, they belong to prokaryotes. Prokaryotes are the simplest organism on the simplest living organism that are present on Earth. They don't have well-defined membranes. Their nuclear material is directly suspended in a particular place in cytoplasm. So you can see that bacteria and archaea that were supposed to be the same in the near past, before I think before 2000, they have been found as distant from each other as between bacteria and eukarya. So bacteria and archaea are quite distant, and they are distantly placed. They are genetically different. The archaea occur more commonly in the extreme conditions, such as in the salt affected areas, or in the hot springs, or in deep sea environment, etc. So prokaryotes are of different types in soil. For example, they are autotrophic prokaryotes, there are heterotrophic prokaryotes, there are saprophytic prokaryotes. But in soil, when we talk, we mostly talk about the heterotrophic prokaryotes. These are the prokaryotes that can utilize the organic matter for their energy purpose. They depend on the consumption of soil organic matter. They are important part of the decomposer community. Most of the energy flow in soil depends on their heterotrophic activity. Basically, they are important part of all the important nutrient cycles that are working, they are functioning in the soil, including carbon cycle, nitrogen cycle, including availability of phosphorus in the soil. Similarly, sulfur reactions and all other things like when the dead organic matter is added in soil, then these are the prokaryotes which play a very important role in degrading all that organic matter and converting them back into their simpler forms that are either utilized by other organisms or that become available again. So these then there is another division in prokaryotes, that is because when we talk about the study of prokaryotes, then we are talking about whether they are culturable or not culturable. The culturable ones are the prokaryotes that can be cultured on plates in the laboratory. The non-culturable prokaryotes are the prokaryotes that cannot be cultured, or they don't appear on the plates. So it doesn't mean that they are not I mean they are not heterotrophic. They are also heterotrophic. And another important thing about these two categories, culturable and non-culturable, it is very interesting that only 1% of the soil bacteria are culturable. So we would be discussing all this in our lecture, that why such a small quantity of bacteria are culturable and 99% of bacteria are not culturable. So how do we find that only 1% bacteria are culturable? And how we conclude about that, why most of the bacteria are not culturable? What are those bacteria which are not culturable? What is their contribution towards the soil soil metabolic reactions or total soil metabolism? So the culturable soil bacteria are those bacteria which are actually that can be cultured on plates. Again, as I already talked about that question, why there is such a small percentage of bacteria that is culturable? There are a few important facts about that. So we can find probably some rationale in that. First is the bias in the methodology. That can be the reason that some bacteria are cultured and we can see them on plates, while others we don't see. For example, we use some culture media in the plates. Again, all the culture media are different from each other, and this is of course a kind of biasness for the bacteria. Some bacteria may better grow in some particular culture media and others may not. Similarly, the incubation conditions. The incubation conditions vary, and they also vary for as far as the bacterial preference is concerned. And then there is a big diversity of bacteria in soil, not only based on morphology but also on physiology. So it's very difficult to pick all of them on plates because sometimes they look very similar and their physiology is slightly different. Then again, the same reason, huge population densities. And the classical taxonomic techniques are very difficult to apply. Classical taxonomy techniques are dependent on some limited characters that are based on either morphology or phenotype or physiology of the bacteria. So similarly, some bacteria grow faster than others, and in this way they appear on plates, while others we don't see on the plates. So the major classes of bacteria found in soil based on classical taxonomy are the Arthrobacter that have been found up to 60%, then Bacillus up to 67%, and Pseudomonas, then Agrobacterium, alkali genes, Flavobacterium. So all this composition you see is very variable from one soil to another soil because of big variability within soils, among soils, and also because of the culture media, culture conditions that are too much selective in picking up kind of population out of the big diversity present in the soil. The culture conditions are, I mean, the media. If you change a media for same soil, you would see different colonies. So media is playing a very important role in selecting which bacteria will be more abundant in a particular test. Similarly, if the soil is coming from forest, or if the soil is coming from grassland, it will contain very huge numbers of different types of bacteria that will be different as compared to the ones coming from a desert or from a culturing from some other type of soil. So soil type is also an important factor because it determines the type of nutrition available to the bacteria, and it also determines the amount of moisture that may be available in the environment. So all these factors affect the type of bacteria that are viable in those conditions.